So we've been looking at red blood cells, erythrocytes, and kind of seeing what's going on with those. Uh, and what we're going to see here is the last thing we're going to talk about is blood types. So blood types are a characteristic of red blood cells, and they're going to be important for a number of reasons, as we're going to see in a little bit. But mainly, it's really about determining what is foreign to a body and what is not. And again, the biggest issue with this is trying to figure out, in some level, the ability to do blood transfusions for need when that need arises. So human blood groups, really it wasn't until about the early 1900s that these were actually identified as being present. It's not to say that they didn't try to do transfusions before that. A lot of times what they would try to do is they might try to do a transfusion every once in a while. It worked great, the person would survive. Other times they would transfuse blood into somebody and they would die quicker than they would have thought they should have. Like I said, it was Carl Landstein in 1900, he won the Nobel Prize for it in the 1930s, that identified one set of the blood groups. There is a lot of other blood types and groups identified, but what we're really going to talk about is the ones, the ABO blood group as well as the Rh factor, these ones that can actually lead to transfusion reactions. Those are the ones that are honestly probably the most key to us. So blood type is determined by these glycoproteins that are on the surface of red blood cells. We refer to them as antigens in this case. And these are genetically based, and any particular person, all the red blood cells in that individual are going to carry the same antigen. So if you have a particular gene, all your red blood cells are going to have that. So with the ABO blood groups, and that's what we're going to start out with here, just the ABO blood groups, and we'll deal with Rh factor. ABO blood groups, type A blood has the antigen that we've named A. Type B has the antigen that we've named B. Type O does not have either the A or B antigen, and what's interesting with the ABO blood groups and the genetics on these ones, there is no dominant and recessive. These are codominant genes. So if you get a gene for A from one parent and a gene from B from the other one, you will actually express both and have both the A and B antigen on the surface, and that ends up leading to type AB blood. One of the things that's interesting with this is we do make antibodies against the antigens that we don't have. There's other proteins and all that is present in nature that generally pretty soon after birth we're going to have antibodies against the antigens that we don't have. So our red blood cells that are type A have A antigen, but what we shouldn't see is we shouldn't see any B antigen. So the plasma of these folks will have antibodies against B. So they have anti-B antibodies. And this can become an issue if B blood cells are introduced to this plasma in these folks. Antibodies have two little branches on them. They have two antigen binding sites. So what this means is any particular antibody can bind multiple antigens. And what you can do is you can start getting this clumping, or agglutination is actually the term that we use on this one. This agglutination is the combining of these red blood cells and antibodies that is going to make this clump. And if you get enough of this blood mixing together, these can get very large. So this type A plasma with type B red blood cells, you're going to get all these starting to group together. If you were to accidentally transfuse a patient, or type A blood, let's say, into a type B recipient, there would be this cross reaction, and what happens is these agglutination reaction, it gets these large clumps, much like a clot going through the blood, even though it's not technically a clot, it's an agglutination. But what starts to happen is you can start blocking blood vessels. So a lot of times branching and smaller blood vessels will start getting blocked by these red blood cells that are all sticking together with antibodies. And this will lead to a blockage here. If you get a blockage in certain areas, this can lead to heart attacks, strokes, many other things like that, loss of blood flow to an area, hypoxia, cell death. And honestly, if you get enough blood, this can lead to overall death. So type B, not surprisingly, B-type antigens, they have antibodies in their plasma against A, because again, there shouldn't be any A antigens there. Same thing here. If we somehow gave A blood, those anti-A antibodies would bind it up. Type O does not have the A or B antigen on the surface of its cell. Therefore, you really shouldn't see either A or B antigens in the bloodstream there, so they have antibodies against both. What this means is somebody with type O blood can only get type O blood cells from somebody else. So they have antibodies against both A and B. So either A blood, B blood, or AB blood would all lead to an agglutination reaction. And type AB blood, we know the antigens are what? Antigens are A and B. Antibodies, well, if you have both the A antigen and the B antigen on your blood cells, you shouldn't have antibodies against either antigen. So what you're going to see is type AB blood has antigens A and B on the surface and no antibodies against either A or B. And what this really means is type AB blood could receive blood cells of any of the other types because they have no antibodies to actually react with it. So this is, again, a nice little summary showing you type A blood. Antigen A on the surface, antibodies against B in the plasma. Type B is the opposite. Antigen B on the red blood cell, antibodies against A in the plasma. Type O has no antigens on the red blood cells, has antibodies against both A and B in the plasma. And finally, type AB blood has both A and B antigens on the red blood cells. Because of that, no antibodies in the plasma. So what we're going to see here is, and this is where it becomes important, over time we figured out that we can actually transfuse blood from a recipient to a recipient from a donor, and if this person is losing blood, possibly save their lives by getting this blood in there so they don't lose too much blood. So this is kind of showing you this blood match type, and I just want to say this is just ABO blood groups right now, no RH factor, no plus or minus, positive or negative, don't worry about that yet. But you can kind of see here, a lot of these make sense. If the recipient is A, you can definitely give A to that person. If the recipient is B, you can definitely give B to that person. If the recipient is O, you can give O for sure. And if the recipient is AB, you can definitely give them AB. So then if we start looking at some of these other ones here, we have a lot of yes question marks. Why is it a yes question mark? This is where some of this gets interesting. Generally, how we give blood donation now is when you go and donate blood, they are going to spin down, they take your blood, once it gets in a little baggie, they were actually going to separate it into what we call packed cells. They will remove the formed elements and put those reconstituted in a bag, as well as actually separate some of that plasma out. If we're talking about whole blood, there could be an issue. So let's do A, for example. Recipient is A. We know we can give it A. Could we give it O? Well, let's think here. A has antigen A on the surface. O cells have no antigens. We know we have antibodies against B in this blood. There's no B antigen on here. If we gave cells from type O to a recipient of A, that should be fine. Why the question mark is there is if you're actually giving whole blood, you're giving both cells as well as the plasma. What is in O-type plasma? You're going to have antibodies against both A and B. If you give too much donor blood from O, the antibodies that are coming in that, if you're giving a whole blood donation, the antibodies that are in the plasma here can start reacting with the recipient's blood. So these question mark ones here, if you're just giving packed cells, that is an okay donation. So A can get packed cells from O. So O is a universal donor. You can give it to all the other blood types if it's just O cells. O plasma, you can only give to O recipients. But again, if you're giving just the cells because there's no antigens on those and you're not giving any of the plasma with the antibodies, that would be a yes. Same thing with AB being a universal recipient. If you're getting packed cells from A going into an AB person, that's fine. But if you're getting A plasma, there's antibodies against B, those can react. So again, packed cell donations, all these question marks become a yes. Whole blood, they are a no. So again, if you're doing a whole blood donation, it should only be type-specific blood donation. The reason I go through this, and it's just because of the simple fact, a lot of people are going into medical fields, and a lot of times after you go through this enough, you'll get into your head that, okay, O negative is the universal recipient, donor. AB positive is the universal recipient. That is true for the most case. If you watch enough shows on TV, they'll say anything else. They usually say spike O-neg, get a bag of O-neg. The reason I say and go through this about the whole blood versus packed cells is just in case there is ever another emergency like that, where like there was that 9/11 where they were triaging people and doing blood donations from one person to another right at a firehouse because there was no hospital beds. If you're ever getting a triage situation, understanding how these work could save somebody's life in that sense. And that's why I do go through these a little bit like that. But that is the issue with these ones. If you're giving enough donor blood with plasma, if you're giving whole blood, it can become an issue. So that is that idea here. So this case study is kind of saying if a person lost 50% of their A-type blood, only O-type blood available for transfusion, could you give this? Yes, if it's packed cells. No, if it's whole blood, because that would be too much plasma and there would be a reaction. The other thing we need to talk about is what is referred to as Rh factor. So Rh factor, it actually comes from rhesus monkeys. It was where this was discovered, but humans have this as well. If you have Rh antigens on the surface of your red blood cells, you are Rh positive. If you do not have Rh antigens on the surface of your red blood cells, you are Rh negative. This is about 85% of humans are Rh positive. So there's not a lot of negative, honestly, on these ones. So Rh negative folks, do they have antibodies against Rh positive? The simple answer to that one is maybe. So this is a little bit different than the ABO blood groups in that to really make, for somebody that's Rh negative, to make antibodies against the Rh antigen, they need to be exposed to the Rh antigen. So if you are Rh negative and you are exposed to Rh positive blood, you will make antibodies against that, and you'll be what we call sensitized, and you will always have antibodies against the Rh factor. If you are an Rh negative person and you've never been exposed to Rh positive blood, you will not have antibodies in your blood against the Rh factor. The presence or absence of antibodies does not change the blood type. Just to be clear, it doesn't change the blood type at all. It just means you're either sensitized or you're not. If you are Rh negative, you are always going to be Rh negative regardless if you're making antibodies against the Rh factor or not. Because again, the blood type is determined by what is on your red blood cells, not the antibodies that are in your plasma. So if you are A negative like this person here, you may or may not be making antibodies against the Rh factor, depending on if you've been exposed to Rh positive blood or you have not. Where this becomes an issue, and again, this obviously if you got a transfusion, you could be exposed. But most folks, if you cut yourself, other stuff like that, you're generally not getting other people's blood in your body. Where this becomes an issue is an Rh negative mother and an Rh positive fetus. During childbirth, when the placenta tears away, maternal blood and fetal blood are going to mix. There's going to be an exposure to the blood antigen of the fetus, and this can lead to some issues possibly. This is part of the reason that back in the day when my parents were getting married, part of getting a marriage license was getting blood typed. And they would talk about this one. Nowadays, they actually give something called, if you're an Rh negative mother, they give you something called RhoGAM. And we'll talk about what RhoGAM is. But first pregnancy here, let's say you have an Rh negative mother and an Rh positive fetus. Generally, the mother is not going to be exposed until childbirth. But when that placenta tears away, what's going to happen is the Rh antigen containing blood is going to mix with the maternal blood. It is going to trigger an immune response. You are going to start making antibodies against the Rh factor. So now this mother is going to have these Rh antigens, antibodies, or glutenins in her bloodstream. If she were to have an Rh negative fetus, this isn't an issue. There's no antibody. These antibodies can't attack an antigen that's not there. But when you get a second Rh positive fetus, there is certain antibodies that can cross the placenta. So what they end up happening on this is if you were to get sensitized, the mother was to get sensitized, has a second Rh positive fetus, her antibodies are going to cross the placenta and start messing with the red blood cells of the fetus. This can lead to stillbirth or more likely a baby born with severe anemia, which is not going to be a good thing either. What they do nowadays is if a mother is Rh negative, prior to the pregnancy, they are going to give a shot of RhoGAM. After the pregnancy, they will type the pregnancy, the fetus, now infant. If that infant was Rh positive, they give another dose of the RhoGAM. So you might be going, what is RhoGAM? And this is where it's really kind of interesting, and I miss not being able to do this in class because everybody kind of gives me that, like, what? Look about it. Kind of that what I was kind of call the puppy dog look, where you make a noise and your dog goes. They give the mother antibodies against the Rh factor. That's what RhoGAM is. So you might be kind of going, they're giving the mother exactly what they don't want her to make. So you don't want the mother to make Rh positive antibodies against the Rh factor. So we inject the mother with antibodies against the Rh factor. Why this works is because, first off, the level of antibodies that you're going to inject, it is a certain amount, and it's a low enough amount that is not going to affect the fetus very much. But what you're trying to do is put enough antibodies in there is that if that antigen during birth was to get into the bloodstream, that there's enough antibodies in the bloodstream to neutralize that and not allow it to trigger an immune response. So basically what we're trying to do is stop the mother from actually getting sensitized by actually neutralizing any antigen that gets in. And that's why they put a certain amount in there. And the main thing with this one is if you just inject those, you haven't had an immune response. The mother's not going to continue to make these. And after about 6 months, those antibodies will be cleared from the mother's system, so there's nothing in there at this point to be an issue. If the mother actually gets sensitized, there is immune memory in there, and she's going to continue to make these antibodies for the rest of her life, and every positive fetus after that point could have an issue. And once she's sensitized, RhoGAM doesn't really do anything to stop this. So if the mother actually gets sensitized, putting these Rh antibodies in there is not going to do anything to help the fetus. So what we do now is anytime there's an Rh negative mother, we give her these antibodies prior to birth as well as after birth to try to protect against any sensitization to that Rh factor. And again, with blood typing, it's the same idea here. You're trying to match the specific type. So you don't give somebody who is Rh negative, Rh positive blood because you'll sensitize them. Somebody who's Rh positive could get Rh negative. But again, as much as possible, we're going to try to type match because, again, that is the best way to do this. If we have time, we always want to do the exact same blood type. So if you're O negative like I am, well, O negative can only get O negative. But if you're like AB or A positive, they're going to try to give you A positive, not an A negative blood. You could get A negative blood. You could get O negative blood. And those are some of the things I want you to think about as we go through these ones. You're going to do a little thing in the lab here that it's going to be doing some blood typing. Understand, because it's a really easy question to ask. If you have a particular patient and I said, you have these types of blood available, what blood could you give to which patient? You should be able to understand these transfusion reactions. If you end up having any questions on them, please ask in any of our live meets about this. So I will see you next time.